So the purpose today is to compare and contrast the trip operon with the lac operon, specifically in prokaryotes. Eukaryotes, of course, being a little more advanced, having cell organelles, undergo very similar types of gene regulation. However, there is a lot more steps involved and a lot more proteins. So if you think about the bare bones of gene regulation in eukaryotes, we're certainly learning very similar ways it's done in the prokaryotes. So what we're learning today about prokaryotes, at least comparing and contrasting these operons, we're essentially learning about the basics of how eukaryotes do it. Eukaryotes, again, a little more complicated. They have cell organelles, so there's going to be a lot more proteins and cell-to-cell recognition things that have to go on there. So again, let's start with these two. And I believe by comparing and contrasting these, we better understand the differences and the uniqueness of them. Okay, so the first thing I have to realize in the trip operon is that essentially I've got some DNA, and in this DNA, okay, upstream, I've got some regulatory places. So I've got some places in my DNA strand that code for the regulatory gene, okay? And of course, then eventually I have the promoter region, and of course, that's where the DNA polymerase would bind. And right next to it, I have the operator. And of course, the operator is essentially the switch that turns on what we call the entire sequence, the transcription sequence that makes the bunch of genes. Now, for the trip operon, there's 5 exon units that will eventually be transcribed, and they make 5 different genes. I'm just drawing some here. So there's essentially 5 parts or 5 different proteins being made, okay? So we need the DNA polymerase essentially to bind, okay, onto the promoter region, and we learned there's a TATA box in this area, and there's some transcription factors that help the DNA bind, and we're kind of going to learn something about these. So this is the RNA polymerase, okay? And when it binds here, it's going to keep, and if there's nothing in its way, it's going to keep going and transcribing messenger RNA. And that messenger RNA, okay, will lead to translation of the enzymes. Now, the enzymes that are made in the trip operon are the enzymes that make, okay, they make tryptophan. Tryptophan is an important amino acid for many different proteins or catalysts that are needed. So let's get rid of this, okay, and understand that this pathway is making. Now, if we're going to make something, okay, we're going to take, we're going to go this way, and we're going to make tryptophan, and we do so by making enzymes, as I just said, that do what? Help take resources, okay, that we have in the cell. So we take resources, and from these resources and with the enzymes, we make tryptophan, which can be used for whatever purpose it's needed for. So we're going to take up some resources. Now we call this pathway, when we make something, okay, anabolic. Very important. Anabolic means we are going to process something. We're going to take something and we're going to build it. And we're building tryptophan from resources, and we need these protein enzymes to be coded to do so. Now, if you're going to build something, okay, it would make sense that we only build it when we need it. That's very important to have that kind of metabolic control, because we need our organisms to be what? Very, very efficient. So we only make tryptophan when we need it. Now, think about this, because people get confused. When I memorize the trip operon, is it always on or is it always off? Well, if we're going to make enzymes to make tryptophan, we only want this to be on when we need it. So if you don't have any tryptophan in the vicinity of this gene, and again, this is in prokaryotes, so there's no cell organelles, so if there isn't any tryptophan, that means you need to make it. So this gene is going to be on, okay? And what I mean by on is there's going to be nothing bound to the operator. So RNA polymerase is going to be able to bind to the promoter, which it always would, but will not be blocked because nothing is binding to the operator. So this would go forward and make the enzymes. So this makes sense that when you are dealing with anabolic operons, most of the time they're on position until otherwise noted. Why? Well, if I'm going to make tryptophan, therefore, I need to be told when I have tryptophan to stop. So when you have tryptophan that comes into play, that is due to, guess what? Not needing it. So when tryptophan starts building up, because you don't need it anymore, why would we keep making more enzymes and using up more resources that takes stuff away from other things that we need in the cell to make tryptophan if we have it? So when tryptophan starts showing up at our door, okay, it somehow signals to stop making the enzymes to make more. It's an incredible good feedback loop, okay? So what happens is tryptophan binds to a regulatory gene we call is a repressor, and that repressor is already there hanging out. So I'll just draw that repressor as a red, let's say, big circle here. So we have these repressors. They're already being made. They're made in low numbers, okay? But because they're an allosteric protein, which means they have a certain shape they can change into and out of, an inactive site and an active site, remember, form and function with proteins, all about their three-dimensionality, they cannot, okay, bind to the operator initially. So they're made in the inactive form. And it's not because I know this, it's because it has to be to explain how this works. If I'm going to make something and I know that the cell is going to work by the premise that it's only going to make and use up important resources that could be used for something else in the life and the times of a bacteria, that it's only going to do so if there isn't any tryptophan. So initially, if there's no tryptophan, it should be the on position. Therefore, the repressor protein, okay, is made and it has no ability to attach to the operator and block the RNA polymerase from transcribing messenger RNA that eventually makes enzymes. But if you have tryptophan building up, that would mean that you don't need it anymore because it's being used for something else and there's plenty of it. Then there's no way we should be still making the enzymes and using up valuable resources. And what happens is the tryptophan binds with this allosteric repressor protein, changes its shape so that now it can bind with the operator. So when this binds, and you should understand what's happening here, probably the tryptophan attaches or is attracted to some of the R groups, okay, that may or may not be exposed here of this quaternary type structure protein. And by attracting some of them, this protein probably refolds into a different shape. And doing so, it now can attach to the three-dimensionality of the operator. The operator has certainly a nucleotide sequence, but it again only attaches to a certain three-dimensional shape. So the tryptophan, when it builds up, signals this operon, okay, which is basically what? One promoter, the operator, and all the genes that make up all the different proteins that do one job, an efficient structure here, signals it to turn off by binding to the repressor protein. It's already being made, and that, of course, allows it to take on the operator. And then once it binds to the operator, it's a physical being that stops the RNA polymerase by going forward. The RNA polymerase cannot go forward once this binds. So that's how I think about it. Okay, now, what about the lac operon? Lac operon is the same idea. You've got DNA, you've got upstream some regulatory proteins, you've got a promoter, okay, place where the RNA polymerase wants to attach. You've got the operator, okay, which is the switch. And then we have the segments of gene that code for 3 different proteins that make the important enzymes to produce the enzymes needed to break down lactose. So the lac operon, okay, what is its job? Well, we would say that it's there to break down the sugar lactose. So it's there to break something down. We call that a catabolic pathway. What's the difference here? Well, think about it. If you're going to break down lactose, why would you keep this operon on unless you have lactose? So think about that. Why would I code for these genes and transcribe these genes to break down unless I have lactose? It would be a waste of resources to take all of the amino acids and put them together to make these 3 what? Possibly enzymes or a complete enzyme to break lactose down, okay, if I do not have to. So this operon, okay, is going to be in the off position, because if I'm going to break something down, this gene or this promoter or this operon shouldn't be on until I see some lactose. That's the difference. In the trip operon, you were making tryptophan. So you only stop making the enzymes for tryptophan when you see tryptophan building up. So therefore, this gene is on to keep going until tryptophan shows up. In this case, because we're making the enzymes to break down lactose, this gene, okay, we need these enzymes, of course. This gene is going to be turned off until we have lactose present. Why would we make the enzymes to break down lactose if there isn't any lactose? So what we probably have learned, if there's lactose present to be metabolized, there's something called allotose, which is basically an isomer of lactose that's made alongside lactose. So you really can't have one in your system without the other. So if we've got lactose present, allolactose is always there. And allolactose is what we call an inducer. Inducer induces this gene to be on. So just like before in the regulatory upstream, in the part upstream from the promoter, where we have the gene regulation or the regulatory genes, we make a repressor protein. And again, that's why both of these operons are on negative control, because they make regulatory genes that can repress and bind to the operator. The difference here is that this one's going to be in the off state. So the lac operon produces an active repressor that has the right shape that can bind with the operator and therefore stop DNA polymerase from going forward. It's a physicality. So in this case, I need to have the lac operon off. Why? Why would I make enzymes and take up resources to make enzymes if there is no lactose to break down? So to be in the off position would mean the repressor has to bind with the operator. Okay? And that really explains the differences. Now, what's nice about the lac operon is we're going to learn you can have some positive control. You can make some other type of upstream regulatory proteins like cap proteins, and these cap proteins are almost always going to be in the inactive site, and they wait for a metabolite, usually something from the cell that indicates that something's not working or a different pathway of energy is being used. One of those metabolites is called cyclic AMP, or cyclic adenosine monophosphate, that when it builds up, it will bind with the cap protein, activate its shape, and the cap protein will bind itself to the promoter region. And what this does is allow the RNA polymerase increases its affinity and allows it to attach here. This is positive control because by having this metabolite activating the camp, it doesn't turn the gene on. Once the gene is on by the inducer, okay, shutting down the repressor and having this operator open, what it does is it kind of like a volume control. It allows the cap to bind here, and it makes it easier for the RNA polymerase to bind. So by having more caps that are activated by the metabolites somewhere in the cell telling you it's doing a different structure of energy, what you're doing is you're increasing the affinity, making the RNA polymerase bind. So the gene is on if the inducer, again, shuts down the repressor protein, change its allosteric shape, but the caps make it increase the rate of the DNA polymerase. So that's the L extra that you can have in lac operon. But if you can understand both, how they differ, the core of understanding is this: the trip operon, you're making something. It's anabolic. And you, again, if you're going to make something, you're going to keep making it because you need it. How do you know if you don't need it? It's going to start backing up on you. And if it starts backing up, this negative feedback loop is going to shut it down. If you're going to break something down, like a catabolic pathway, well, you're going to make and use up resources to make the enzymes only if it's present. And that means, okay, this gene is off until you see what you need. This gene is on until you see something backing up. That means I don't have to make it. Hopefully that's clear enough. Good luck.